Hi, I want to introduce you to someone. This is Ted. Ted is 85 years old, still practices as a patent attorney, has 5 kids and 9 grandchildren, and I'm one of them. So yes, this is Ted Zalenski, but to me, he's Jaja, which is Polish for grandpa. About 10 years ago, my grandpa was diagnosed with leukemia, and as a family, we were incredibly concerned and worried and just wanting him to get the best treatment he could. But unfortunately, he wasn't responding to his treatment. But luckily, he was enrolled in a clinical trial at Ohio State, which saved his life. As a family member, I am incredibly grateful, as we all were, for this treatment to be afforded to my grandpa. But now, as a cancer researcher, I'm even more inspired by this story, and I'm motivated to hopefully someday do this for another patient and their family. So I don't study leukemia, but I do study another type of cancer called glioblastoma. Glioblastoma is the most lethal primary brain tumor with an average survival of 12 to 14 months after diagnosis. The standard of care for glioblastoma is surgery, followed by radiation and chemotherapy. And this hasn't changed in over 15 years, despite the 5-year survival rate being only 5%. And this is due to the fact that recurrence of these tumors is nearly universal, even given our best treatment efforts. As someone who studies this tumor type, I find this unacceptable. And I find it devastating that this is what we tell patients when they're diagnosed with glioblastoma. But I do think there are things we can do. So to understand the shortcomings of our current treatments, we need to understand where they are failing. And something that I'm working on studying is the cancer stem cell population within glioblastoma. Cancer stem cells are a particularly lethal subpopulation of cells within glioblastoma, and they're lethal for a couple different reasons. One being that they're a very invasive cell type, and they migrate away from the bulk tumor, as you can see here with the blue cells. When a surgeon goes in and removes the bulk tumor, they're only going to get that tumor that they can see that's still in a bulk mass, not the tumor cells that have migrated away. So that's why we have our second line of defense, radiotherapy and chemotherapy. But unfortunately, cancer stem cells are chemoresistant and they're radioresistant. So as you might be able to tell, these cells are just left behind and they stay in the brain. And this really becomes a problem. So we have these cells that get left behind. We have both cancer stem cells and non-cancer stem cells. So we come in with our second line of defense of radiation and chemotherapy. And that actually does a pretty good job at killing the non-stem tumor cells that are left behind, because they can also migrate away from the bulk tumor. But it doesn't do anything for the cancer stem cell population. So these get left behind, and ultimately, they're able to regrow the tumor. And this is what drives recurrence and ultimately leads to patients succumbing to their disease. So hopefully it's obvious that we need to inclusively target the cancer stem cell population to effectively treat glioblastoma. And one way I'm looking at doing that is by targeting a protein called KIF11. KIF11 is a protein that is involved in cell division, and it's absolutely critical for cells to progress through mitosis. Not only this, but it's overexpressed in GBM cancer stem cells. So if you can think back to high school biology, you may remember seeing this graphic of how cells proceed through mitosis. But what you might not know is that KIF11 is absolutely required for this to faithfully occur. When we apply a KIF11 inhibitor, cells are stuck in mitosis or they will die, shown in the bottom graphic. So knowing this, we thought that KIF11 inhibitors might be a useful strategy to try and inhibit cancer stem cell growth and eventually kill cancer stem cells in glioblastoma. So we tried this in our lab, and in a laboratory model of GBM, we did see that a KIF11 inhibitor was able to prolong survival, but the tumors did recur when treatment was stopped. And so if you can think back to that graphic, they were getting stuck in mitosis, but the cells weren't dying. They were able to keep growing after treatment was stopped. And this is a bit of an issue, but we think we can improve upon this. So this is where my project begins. I'm working with a KIF11 inhibitor, but also using, in combination, radiotherapy. And the reason this works is because KIF11 inhibitors, again, arrest cells in mitosis, as shown here. So we'll pre-treat with the KIF11 inhibitor. Then, when the cells are arrested in mitosis, we'll hit with radiotherapy. And this is critical, because when cells are arrested in mitosis, it is well demonstrated through years of research that this is the phase of cell cycle where they are most sensitive to radiotherapy. So we're hoping that by pre-treating them and getting them stuck in mitosis, we'll be able to increase the efficacy of radiotherapy, not only to kill the non-stem tumor cells, but also to inclusively target and kill the cancer stem cell population. So we've begun studying this in our lab, and how we're doing it is taking patient-derived specimens and growing the cells and then treating with ARI-520, which is a KIF11 inhibitor, and then following that shortly thereafter with radiation. We then put the cells on an incubator with a microscope to monitor them and see their growth over time. And we have some pretty promising results. So this first patient specimen, called NU757, you can see in the red, this is the untreated cell population, and those grow very well. And then the dark blue and the pink are each treatment used singularly. It stops growth a little bit, but not perfectly. And lastly, we have the light blue line, which are the treatments used in combination. And you can see that those cells grow the least, which is exactly what we want to see. We also see it here in another patient specimen, 3691. And this is even a little more striking. Again, we see the blue line basically having almost no growth over time. And so these are really promising results that combining KIF11 inhibitors with radiotherapy is a viable therapeutic option for glioblastoma. We're very excited to move into new models and to keep testing this to hopefully we get to a point where this could benefit glioblastoma patients. So to close, this is me and my grandpa at my wedding last weekend. My grandpa was not only able to attend, but he did a reading at the ceremony, he spent time with family, and he was able to even join us on the dance floor. This is what I want for glioblastoma patients. I don't want their diagnosis to be a death sentence anymore. I want them to be able to live life and do the things that they want to do. And I'm hopeful that by using KIF11 inhibitors combined with radiotherapy, we might be able to do that. Thank you.